Good morning, everyone. And OIS, thanks for having us. This is Annexon Science's first visit to OIS and presentation this morning, and we're happy to be here. All righty, we're a public company. We'll be making forward-looking statements, and we refer you to our documents on file. So, Annexon, who are we? We are focused on the classical complement pathway and stopping the disease processes and the indications in which we are pursuing right at the start. We are indeed pursuing two distinct disease processes with our approach. The first is antibody-mediated autoimmune disorders, very well known for the role of complement there. The second is what we call complement-mediated neurodegeneration. This was discovered by our scientific co-founder, the late Dr. Ben Barres, former chair of neurobiology at Stanford, who discovered the role of C1q in removing functioning synapses in the brain, as well as functioning retina ganglion cells and photoreceptor cells in the eye. As a result, we have a platform approach across three therapeutic areas in the autoimmune, neurodegenerative, and ophthalmic space, with three clinical stage drug candidates, advancing currently 5 phase 2/3 clinical studies, 2 on the horizon, and anticipate 7 clinical data readouts over the next 2 years. Our mission plainly is to bring disease-modifying therapies for patients suffering classical complement-mediated disorders. Brief snapshot of our pipeline focused on the ophthalmic approach. ANX007 is our lead drug candidate there. It is a monoclonal antibody Fab fragment for intravitreal administration. We completed a placebo-controlled phase 1b study in patients there where we demonstrated it was safe with full target engagement, and we'll show you some data on that. So why C1q in the classical pathway? Well, in short, for diseases for which we believe complement is the key driver of the process, we're providing more complete protection by blocking C1q right at the start of the pathway. Indeed, you are likely all very familiar with complement and the enzymatic amplification of the cascade. By blocking C1q, we're blocking all upstream immune cell infiltration and attack that results in inflammation and tissue damage in a host of diseases. That thus gives us broad therapeutic potential across a range of diseases, and we'll show you some data as it relates to that. Just a snapshot in some of the indications for which we've produced preclinical data in multiple different models in multiple different labs, frankly, around the world. Here on the left are a couple of models, distinct models, one in acute glaucoma, the second in chronic glaucoma. And there you can see by inhibiting C1q, we're able to preserve optic nerve damage to optic nerve. And in the second case, as it relates to the chronic study, it was actually done in comparison with C5, where again, we showed more complete protection because we were blocking upstream cellular infiltration and tissue damage. On the right is a third model in optic neuritis, where we blocked or preserved nerve loss by blocking C1q there. Importantly, as we turn to geographic atrophy, we think it's very important to block C1q in the classical cascade right at the start. And there are a couple of reasons for that, one of which is depicted on this slide. C1q is indeed deposited and activated in the key hotspots for the early aspect of the disease process. In the top right box, you see C1q deposited on photoreceptor cells in aged mice. On the bottom, you see C1q deposited on drusen, obviously a key driver of geographic atrophy. Also, this is in patients. And in the middle, you have C4 just downstream of C1q and the activator and key activating component of the cascade deposited on photoreceptor cells, again, in GA patients. So C1q is really at the start of the disease process, and it is activated, and thus we think playing a key role in driving the disease process right at the start. Another differentiator of targeting C1q versus other approaches, of course, is that C1q, C4b, and C3b are separate opsonins which attract macrophages and microglia cells that again result in inflammation and tissue damage upstream. By blocking C3, C5, and downstream, these opsonins remain in place and continue to attract macrophages and microglia activity, which frankly can only be blocked by blocking C1q at the start of the cascade. So as a result, we think we are providing more complete complement protection by blocking the cascade before it gets started right at the start in the disease process. Some data as it relates to geographic atrophy, again, in different mouse models, models that actually have also treated or been used with C3 inhibition as well. There, we've been able to protect photoreceptor cell thickness as well as function. Again, measuring function we think is quite important in showing the utility of blocking C1q. And this data set certainly resembles, if not better, than what we've seen with C3 in this model. As it relates to ANX007, as I said previously, we have treated it in patients in a phase 1b sham-controlled study. The drug was safe and well tolerated, and importantly, it had full target engagement at both the low and high doses for at least 29 days. There, we pulled aqueous humor from patients to assess that engagement. We did not do a second tap, but based on our preclinical package in monkeys, we're fairly confident that we can inhibit C1q monthly and every other month. And that's given us the encouragement to move forward into a geographic atrophy phase 2 study, which is well underway. We're about 50% through the study. This is a sham-controlled study where we are taking 2 doses in monthly, or 1 dose, but a monthly and every other month construct, 12-month treatment, 6-month off treatment. And the primary endpoint, as you would expect, is change in lesions as measured by fundus autofluorescence. As I said, this study is well underway, and we anticipate data in 2023 with regard to that. Finally, looking ahead, we're all in on the ophthalmology franchise. We're doing a significant amount of work, some with many of you in the room here, as well as others, in multiple indications where we think C1q is a key driver of photoreceptor cell loss, plain and simple. So we're started in geographic atrophy. We are shortly beginning to look into intermediate AMD to perhaps prevent blindness earlier in the disease process. We also are looking at inherited retinal diseases where we can preserve the loss of photoreceptor cells earlier. And we have a robust data package in glaucoma. These are progressing glaucoma patients. We've done a lot of work to identify patients who are progressing so that we can run a smart, feasible phase 2 study there and then move forward into phase 3. So I appreciate your time this morning. Thanks so much.